Hi everyone, I am Dr. Shagirtha, Assistant Professor, Department of Biochemistry from St.Joseph College of Arts and Science, Cuddalore. In this video, I am going to discuss about the structure and functions of DNA. The nucleic acid or the organic material present in all organisms in the form of DNA or RNA. These nucleic acids are formed by the combination of nitrogenous bases, sugar molecule and phosphate group that are linked by different bonds in a series of sequences. The DNA structure defines the basic genetic makeup of our body. In fact, it defines the genetic makeup of nearly all life on the earth. First, what is DNA? It is a group of molecules that is responsible for carrying and transmitting the hereditary material or the genetic instruction from parents to offspring. So, it is known as deoxyribonucleic acid. So, most of the DNA is located in cell nucleus with small amount of DNA found in mitochondria. Apart from being responsible for the inheritance of genetic information in all living beings, DNA also play a crucial role in the production of proteins. Nuclear DNA is the DNA contained within the nucleus of every cell in a eukaryotic organism. It codes for the majority of the organism genomes, while the mitochondrial DNA and plasmid DNA handles the rest. The DNA present in the mitochondria of the cell is termed mitochondrial DNA. It is inherited from the mother to the child. In human, there are approximately 16,000 base pairs of mitochondrial DNA. Similarly, plasmid have their own DNA and they play an essential role in photosynthesis. So, we discussed about the types of DNA. There are three different types of DNA: aDNA, bDNA and zDNA. First, we discussed about aDNA. It is a right-handed double helix similar to the bDNA form. Dehydrated DNA takes an A form that protects the DNA during extreme conditions such as the desiccation. Protein binding also removes the solvent from DNA and the DNA takes and form aDNA. Next one is a bDNA. It is the most common DNA. Conformation and its right-handed helix. The majority of DNA has a bead type conformation under normal physiological conditions. Next one is a zDNA. It is a left-handed DNA where the double helix wind to the left in a zigzag pattern. It is discovered by Anthros and Alexander Rich. So, next we are going to discuss about the structure of DNA. It is right-handed double helix with about 10 nucleotide pairs per helical turn. Each spiral strand composed of sugar phosphate backbone and attached bases is connected to the complementary strand by hydrogen bonding between the pairs bases adenine with thymine, guanine with cytosine. So the DNA molecule is composed of units called nucleotides, and each nucleotide is composed of three different components such as sugar, phosphate group and nitrogenous bases. The basic building blocks of DNA are nucleotides. So these four nitrogenous bases, mainly we already discussed about the nitrogenous bases, mainly the adenine with thymine, guanine with the cytosine. These base pairs are essential for the DNA's double helical structure, which resemble a twisted ladder. In order to nitrogenous bases, it determines the genetic code. The genetic code is very important. Among the three components of DNA structure, sugar is the one which forms the backbones of DNA molecule. It is also called deoxyribosugar. The nitrogenous bases of the opposite stand from hydrogen bonds forming the ladder-like structure. So the DNA molecule consists of 4 nitrogenous bases. They are adenine, thymine, cytosine and then guanine. These two strands of DNA is run in opposite direction. These strands are held together by centrally hydrogen bonds that is present between the two complementary bases. The strands are helically twisted, where each strand forms a right-handed coil and 10 nucleotide makes up a single strand. The pitch of each helix is 3.4 nanometer. Hence, the distance between the two base pairs is 0.34 nanometer. The DNA coils up forming chromosomes, and each chromosome has a single molecule of DNA. Overall, human beings have around 23 pairs of chromosomes in the nucleus of cells. DNA also play an essential role in process of the cell division. Next, we are going to discuss about the Chekhov rules. The Chekhov rule is very important. Erwin Chekhov, a biochemist, discovered that the number of nitrogenous bases in the DNA was present in the equal quantities. The amount of adenine is equal to the thymine, where the amount of cytosine is equal to the guanine. In other words, the DNA of any cell from any organism should have 1:1 ratio of purine and pyrimidine bases. Next, we are going to discuss about the DNA replication. The replication is an important process that occurs during the cell division. It is also known as semi-conservative replication, during which DNA makes a copy of itself. The DNA replication takes place in 3 stages. Step 1 initiation, step 2 elongation and step 3 is termination. So last slide we are going to discuss about the functions of DNA. DNA is a genetic material which carry all the hereditary information. Genes are the small segments of DNA consisting mostly of 250 to 2 million base pairs. A gene code for a polypeptide molecule where 3 nitrogenous base sequences stand for 1 amino acid. Polypeptide chains are further folded in secondary, tertiary, quaternary structure to form different proteins. As every organism contains many genes in its DNA, different types of protein can be formed. Proteins are the main functional and structural molecule in most organisms. Apart from storing genetic information, DNA is involved in replication process, mutation, transcription, cellular metabolism, DNA fingerprinting and gene therapy. In this replication process, the transforming the genetic information from one cell to its daughters and from one generation to the next and equal distribution of DNA during the cell division. Next, in mutation, the changes which occurs in the DNA sequences. Next one is a transcription, metabolism, DNA fingerprinting and gene therapy. Thank you.